In this tutorial, we shall deal with cell-cell interaction, which is here shown as to be subdivided into three subcategories: cytoadherence, invasion, and motility. We go first to cytoadherence, and we see here several maps, and we'll go first to the interaction between the modified host cell and the endothelial cell. We see here the membrane of the infected cell with various parasite protein embedded into them, and here the cytoskeleton of the erythrocyte, which is interacting with different parasite protein, and these are the different receptors on the endothelial cells. We go back to cytoadherence, and we have here a very interesting, valuable table that displays all the proteins that are exported from the parasite and that remodel the infected erythrocyte. And some of them are, of course, also important for cytoadherence. Another hallmark of cytoadherence is the rosette formation between normal and infected erythrocyte. Here shown are the different ligands on the erythrocyte surface, the infected erythrocyte and their receptors on uninfected erythrocyte, with the structure of two of the major players in cytoadherence, the stevo protein and the rifin proteins. Now here we move to invasion. We see several schemes and tables. We'll first go to a famous map by now, the subcellular localization of protein involved in invasion. You see that almost 100 proteins are involved. All of them have been here we see the subcellular location of protein involved in invasion. This is the merozoite with its different subcellular organelles, and all the proteins are shown they are associated either with micronym, ROP3 neck protein, ROP3 protein, surface protein, and so on. You'll see that all of them have the loop icon, which indicates that they have been localized subcellularly. We go back to invasion, and we see that we have a table which not only shows all those proteins but gives also some information about their localization, their potential function, and the features and the structure. Some of the scheme are more detailed. Here is an example of the involvement of the ROP3 neck protein during invasion, and show the various steps of the reorientation of the merozoite toward the host cell membrane, the secretion of the organelle, and essentially the invasion. Now we move to motility, and just following the last dealing with the invasion, we go to components of the linear motor, which is responsible for invasion. We see here the various components, and but this sub-menu includes also other important things like molecular motor prototypes, which show the kinesin, dynein, and myosin, and all their different structures, which are encoded by parasite protein. and this is the example of actin and filament, the formation of the filament, the dissociation and depolymerization of actin, sequestration of monomers, and so on. If we go back, we can see that we have the control of spindle movement by different kinesins, and all of them can be found here.